Okay, so we're on slide 37, linkage analysis, and I will be describing the two main types, which I mentioned previously, parametric and non-parametric. And slide 38 is a definition of parametric linkage analysis. So, as it sounds, parametric linkage analysis involves the assumption of certain parameters, and that makes it model-based. And linkage analysis, parametric linkage analysis, is made possible through the violation of Mendel's law of independent assortment. We are basically testing the hypothesis of no linkage, the null hypothesis of no linkage, which is theta equals 1/2 or 0.5. There are lots of advantages in slide 39 of parametric linkage analysis, and like any statistical model, if it's correct, we can absolutely have increased statistical power for any analysis that would result from a correctly specified model. So if you are lucky enough to specify the correct model, then you have a high probability of detecting evidence for linkage between a marker and a disease gene. And this is why for Mendelian diseases, the approach was so successful. Parametric linkage analysis utilizes every family member's phenotypic information and genotypic information if it's possible. And it does provide an estimate of the recombination fraction, so that's theta. And it also, we've already gone through these formulas, but it provides a statistical test for linkage and for genetic heterogeneity, which we won't go into great detail there, but you can actually test for linkage and make yet one other model assumption, which is that you could actually test for two different loci segregating in a group of families. There are a lot of complications that can be addressed in linkage analysis. As I mentioned previously, I'm interested in the basic understanding and the basic concepts for this course. Okay, slide 40 tells us what's required to do linkage analysis. So, as you know, we need families. Extended pedigrees are terrific. They are much more informative, but we can do linkage analysis with SIB pairs. And we need to have good phenotypic information about the disease. And even though it might seem obvious, it may not be. If you misspecify someone's phenotype in a linkage analysis, it has drastic implications for the resulting LOD score that you might calculate from a particular pedigree. So the phenotype info is really, really important. There's not a lot of room for forgiveness. And we need DNA from family members and genotypes, and then to actually do the analysis. And traditional linkage analysis has been called in the past single point or 2-point analysis. They mean the same thing, and basically what that means is that you are testing for linkage between a genetic marker and a putative yet unknown disease gene. So slide 41 just lists the approach to linkage analysis. If you were actually going to be doing this, you state the components of the model, and I gave you an example of that in the phase known model that we reviewed a few moments ago. So you have to state the components of the model. Is the disease recessive, dominant, rare, fully penetrant, etc. And then you have to assign, like we did, the underlying disease genotypes, given the information about the genetic model. Then you have to determine phase. Then you have to score the meiotic events as recombinant or non-recombinant. And then using the formula that I gave, you would calculate and interpret the LOD score. And when you start adding a lot of families into this type of a mathematical problem, it can become very computationally intensive, And it quickly becomes computationally intensive when you have the situation of phase unknown and you have to start assigning probabilities to the possible numerators for the likelihood ratio. Okay, so slide 42 is just an example from Alzheimer's disease. This was a study that was published long ago, but it was the very, very, very first study that provided the strong linkage evidence for a major genetic effect, which turned out to be the ApoE gene, or the apolipoprotein E gene, for late onset Alzheimer's disease. This is now a very, very well established gene for late onset Alzheimer's disease, but the location of it is on chromosome 19, and it was first identified through linkage analysis back in 1991 in a study of 28 families. And I'm just going to show you results from one microsatellite marker that was used. So all of these families with Alzheimer's disease were carefully characterized for genotypes across the genome, and microsatellite markers, those markers that I mentioned earlier, were used, a whole panel of them across the genome. And this marker, D19S246, refers to a marker that was on chromosome 19. That's what the D19 means. That's the nomenclature for those numbers, or for those markers. The first three refer to the chromosome. And if you go to slide 43, there's a lot of information here, so I'll take you through it. This is the result from doing linkage analysis for all of those 28 pedigrees, families, that had members with Alzheimer's disease in it. And this is the linkage analysis results for that one genetic marker, the D19S246. And the question was, is this marker, are the alleles at this marker, co-segregating with Alzheimer's disease in these pedigrees? So if you look on the left, you will see pedigree in the first column, and every number there, 409, 705, 732, and on, refers to a family. So every line refers to a family, and those data are from each calculation from each family. So the top of this table in red, going across horizontally, shows the possible values for theta. And you'll remember, and this is where it really is important to understand, the reason why you would calculate theta for a large number of pedigrees is so that you could combine them in this way and sum them and then look for the maximum LOD score across the families. And the maximum is basically the biggest LOD score, and it maximizes at the theta value that goes along with it. So why is that interesting? Well, every family could give you a higher LOD score at a particular value of theta. What you really want to know is, taking all of these families into consideration and all the data together, where is the most evidence for the highest LOD score and which theta value corresponds with it. So if you look at this table, every box is a LOD score that was calculated. We're not looking at the pedigrees here. I'm just showing you the final result. And every box, every cell, contains a LOD score that was calculated for the given theta in red above. And if you take all 28 families and all of the LOD scores that were calculated for all the possible theta values that could be looked at, and you sum them and you look at the total at the bottom, you will see that the maximum LOD score is 1.572, so it's greater than 1, not 3, but that's another story. It's greater than 1, 1.572, and it's at theta value of 0.15. So if you go to the next slide, 44, this just shows another way that LOD scores can be plotted. So you can see on this LOD score curve, these are just generic results, where for every value of theta, the resulting LOD score is plotted, and you can quickly see for this example, which is not the one from the previous, this is just another one, you can see where the maximum LOD score is and the value of theta of where it maximizes. Okay, so slide 45, just kind of reminding you of things we've talked about already. So the evidence for linkage, there are criteria. The world really became excited about anything that would show a LOD score of 3 because of the reasons I mentioned. That's equivalent to the data being 1,000 times more likely under linkage than under no linkage. And LOD score of -2 is a critical threshold that was well accepted for declaring no linkage. The problem was for many diseases as we moved forward away from Mendelian diseases into more complex diseases is that we didn't necessarily get LOD scores of 3. And also, one of the biggest problems was that previously we might have had a really strong idea of where a disease gene might be, but when we started screening the genome to look for evidence of linkage, you start really getting into multiple testing issues, which only begin to grow from this point forward in our studies of genetic epi, which you're going to hear lots about as we move forward to using way more than 300 to 400 markers. So there was a lot of discussion in the literature about what would be the appropriate interpretation for LOD score analysis, taking into account multiple testing issues and so forth. And if you go to slide 47, you'll see that one way that scientists decided to interpret results from linkage studies was to say, okay, anything with a LOD score of 3 is definitely evidence for linkage, and please go get confirmation from an independent data set. Above and beyond that, or I guess I should say below and beyond that, a LOD score of 2 was considered very interesting, and a LOD score of 1 was, from a single data set, was considered interesting and worth including in follow-up. So this was the way scientists were interpreting linkage analysis results once we moved to doing whole genome screens for linkage and with many more markers, and also the new understanding that we might not be seeing LOD scores of 3 because the genetic architecture was just starting to be known to scientists to be very different from what people thought would occur from doing linkage analysis in these complex diseases. Okay, so I'm going to skip 48 because I mentioned this previously, that you can actually model genetic heterogeneity in linkage analysis, but just for the purposes of our lecture here, I'm not going to go into great detail. You can read about this in the assigned reading as well. And same with 49, the multi-point LOD score analysis. It's just basically a way where you could extend 2-point analysis to a whole map of markers around a genetic region, and basically using haplotypes to determine LOD scores, and that gave us more information. And this was a really popular way of looking at LOD scores. Okay, I'm going to move on to, again, there's a lot of that in the reading if you want to go into that in more detail. I want to wrap up by talking about non-parametric linkage analysis on slide 50. So this is a less powerful approach compared to the parametric linkage analysis like the LOD score analysis we've been talking about, but the advantages of non-parametric linkage analysis is that you can use affected SIB pairs, so that's a situation where you don't need to have all of the big extended pedigrees, and the statistics are much easier, and so there are some advantages. Less powerful, but definitely a robust alternative. So if we move to slide 51, there are a couple of ways that we can think about sharing alleles, because basically the fundamental concept underlying non-parametric linkage analysis is sharing of alleles at a particular marker locus under investigation. So you can share alleles with someone else in a population, either identical by state or identical by descent. It's IBS and IBD, and these are very important concepts. And so if you go to slide 52, we're going to first start talking about linkage analysis and the kinds of families that you need. So I showed you this slide during the first week of class where we can use affected SIB pairs for linkage analysis. And I showed you slide 54 also. I'm just reviewing where you can determine the possible genotypes for two offspring from this mating. And given that the parents are the father is a 1-2 and the mother is a 3-4 in slide 54, if you take a look at the possible genotypes for offsprings that come from these matings, this is where you can see the identity by descent distribution, and that's expected distribution. We call these expected probabilities for identity by descent, and so the probability of sharing identical by descent or identical by descent is 2. 25% of the time it's 1, 50% of the time, and it's 0, 25% of the time. And you can see those on the right side of this slide. If you look at those, you'll see this is the definition of identical by descent. IBD 0, 1, and 2, those are the expected probabilities from the mating of parents and comparing any two offspring from that mating. You can see in slide 55 that you can actually calculate IBD for other types of relative pairs, and these are the expected probabilities for sharing. So if you take a look on the left, these are the possible different affected relative pairs that you could study. So for example, if you wanted to look at SIBs, those are listed there already. So we know that the expected distribution of sharing between siblings is a quarter, a half, a quarter. But if you go above siblings or below, you'll see the other possible relative pairs that could be interesting if you wanted to look for evidence of sharing. Because basically what you're doing in linkage analysis with relative pairs who are both affected with disease is looking for sharing more often than you would expect by chance. So we expect siblings to share a quarter, a half, a quarter, 0, 1, 2, under the null hypothesis. But if they tended to share more, share 1 or 2 alleles more often than 50% or 25%, then that would be evidence for linkage for a particular marker and disease. So you can do that with other types of relatives like half sibs or cousins or aunt, nephew, grandparent, child, parent, child, etc. Parent, child is another easy one to think about because parents, you share with your child one allele, IBD. So that is something that you can test as well. Any of these distributions could serve as null distributions if you wanted to test for linkage, if you were looking at combinations of these relative pairs who had disease. Okay, so slide 57, just pointing out that IBD status can actually be ambiguous even if you have genotypes from parents. And so if that happens, then again, like I mentioned previously, sometimes you have to assign probabilities to the possibilities here, and that can, for more complicated calculations, you can take that into account. And if you take a look at 58, another way to get around missing parental genotypes is maybe to look at other SIBs. If you happen to have those in a mating, they could help you assign the actual sharing status for other SIBs. So that if you only had the affected SIBs in this example, slide 58, you could study this, then numerous parental mating types would be possible. IBD could be 0, 1, or 2. You just don't have enough information. But if you add the additional SIBs to the picture, then you can infer missing parental genotypes, and then that would allow you to actually assign parental genotypes. And then you could actually say that a SIB pair was sharing 0, 1, or 2. So if you have a situation in SIB pair analysis where you cannot make the assignment of 0, 1, or 2, you can actually assign probabilities to that possibility of 0, 1, or 2. Okay, so I've already said what's in slide 59, just defining IBD versus IBS. IBS, well, I've defined IBD, but IBS is identical by state. So in this situation, pairs can share the same allele regardless of ancestral origin. So unrelated people can share alleles, IBS, and that's slightly different in meaning than IBD. IBD implies that relative pairs have inherited the same ancestral allele, and we can trace that allele from a common ancestor down the family tree to the descendants. So IBD and IBS are types of sharing, and they're very, very useful in determining a null hypothesis, or actually even looking, across populations as well as in families, to generate measures of sharing. We'll see lots of that as the semester goes on. All right, slide 61, of the advantages of affected SIB pair analysis. I alluded to some of these earlier. I think the one that I'll just underscore again that I didn't say was that besides, well, I guess I did mention this a little bit, but affected SIB pairs are easier to collect because you don't need the extended pedigrees, which can be difficult, especially for late onset disorders. A lot of people in bigger pedigrees may have passed away, and unless there was some reason why you would have collected DNA from them, it may not be possible to do a large pedigree analysis. So affected SIB pairs are all from the same generation, and usually you can much more easily find them. You only need the affected individuals in the best case scenario. Of course, as I said on a few slides back, if you have unaffected individuals, they could help you sometimes with assigning IBD status, but conceptually, affected SIB pair analysis is definitely much easier than linkage analysis. The math is easier. And there are a few different affected SIB pair tests, and all of them are pretty straightforward, things that you may be familiar with. So one of them is a chi-square goodness of fit test, where you basically compare the observed proportion of SIB pairs sharing 0, 1, and 2 alleles, IBD, with the expected proportions of a quarter, half, a quarter, under the hypothesis of no linkage between the marker and disease locus. So that's your null hypothesis. That's one way of doing it. So you are doing a chi-square test on the counts, and I'll show you an example of that. You can also just look at a smaller portion of that distribution. You can just compare the proportion of SIB pairs who share exactly 2 alleles, IBD, with the expected proportion of a fourth, 25%. And there is one other way, which should be on this slide. Here it is. That's the means test, where you compare the average IBD sharing in the sample pair with the expected value of 0.5. That's on slide 64. And if you look at slide 65, this is an example of what you would see. So these are real data that came from a publication many years ago, where a group of SIB pairs with type 1 diabetes were studied, and they were genotyped for different genes in the HLA region on chromosome 6, and this is just showing one of those results, simplified. And so there were 137 affected SIB pairs, and the sharing at this particular gene, at an HLA gene, was 10, 46, and 81. That's how the SIB pairs could be categorized according to their sharing. 10 of the pairs shared 0, 46 of them shared 1 allele, all of this is IBD, and 81 of them shared 2. That was the observed distribution, and that should sum to 137. And then the expected comes from the quarter, half, a quarter, which is what the null would be, that we would expect if there was nothing going on and they were just sharing 0, 1, 2 as you would expect under the null hypothesis, you would see 34, 69, 34. Again, that adds up to 137. And so the results of the 3 tests that I just showed you in the previous slides were all highly significant for these data. P-value very, very small, regardless of how you looked at it. If you looked at just comparing the whole distribution 0, 1, 2, or if you just looked at 2, or if you did a means test, whatever. Any way you slice it, the observed data were very different from what was expected, and so the null hypothesis was rejected. All right, so what about when IBD status cannot be determined? So slide 67, inferring IBD from IBS. There are ways you can do this. I'm not going to hold you to too much of this information. I just want you to know that one of the things that I mentioned was that you could just sum over possible mating types to get the IBD probabilities for the affected SIB pairs. So similar to the linkage analysis that I described earlier, you can assign probabilities to possible distributions. Mainly we just want you to know that there are ways to deal with uncertainty in the data, lots of different ways. Again, I just want you to know the basic concepts of affected SIB pair analysis. Now, one last thing I do want you to know is in slide 68. So the problem with affected SIB pair analysis is that you can do those statistical tests and you can determine whether your observed and expected are different from one another with a chi-square test, etc. But you don't end up with a LOD score, and so the true LOD score that you know about, comes from the pedigrees, and the theta is part of that, and so forth. But there was a method put forward by Neil Risch some years back, which was called the maximum LOD score method, and it allowed the results from a SIB pair analysis to be converted easily to a likelihood ratio, which could then be a LOD score. So this MLS, the maximum LOD score, and I want you to know what it is because you will see it in some of the papers that we will look at. It compares the probability of the observed genotypic data in the affected SIB as a function of the proportion of sharing, 0, 1, and 2, with the corresponding probability of the observed marker data under the null. So it's basically a likelihood ratio test using the observed and expected 0, 1, 2 IBD. So let's take a look at a real example, and I think you'll see this is very, very straightforward. So if we look back at that example that I showed originally, you can see how these data can be used to then convert that chi-square table into the maximum LOD score. So this is the log 10 of the likelihood ratio, producing a LOD score the way we produced a LOD score previously for linkage analysis in the extended pedigrees. But here in the numerator, we have the proportions of sharing, 0, 1, and 2, and then taken to the counts, which is the exponents, and then the denominator is the expected proportions, 25%, 50%, 25%, again, with the exponents of the actual counts of the SIB pairs in those categories. So if you look back at slide 65, you will see where those numbers are coming from. So those numbers are coming from 65. The observed was 10, 46, and 81. And if you take a look at this back on slide 69, the 10, 46, 81 is what is being used for the exponents here, for the numerator and the denominator. And the only thing that's different for this likelihood ratio is that we're using the proportion of sharing, which was observed, which is the numerator, and the proportion of sharing, which is the expected IBD, in the denominator. And you can see that produces a very big LOD score, which goes along with that very, very small p-value that we saw. Okay, so slide 70, inferring IBD. There are lots of algorithms that can estimate IBD proportions, and that becomes really complicated, and you do need a lot of computational power if you've got big data sets with lots of affected SIB pairs, and maybe not all of them are can you easily assign 0, 1, 2, so you've got to assign probabilities to what they might be, and you need to do a lot of number crunching to get that. But at the end, once you get your estimates for observed and expected sharing, then you can do your statistical tests, and which is I've just shown you the examples of the statistical tests that you can use, and then also converting it to a log score. And you would be expected to be able to do that by hand for the simplest example. Should not be a problem. Okay, the last thing I want to say then is that slide 71 is uh, referring to quantitative traits. So up to this point, we've been talking all about, I guess, the equivalent of sort of a binary phenotype. You either have the disease or you don't. You can use linkage analysis for quantitative traits as well. And let me just say this was a very big area of method development and research for many years, and quantitative traits are still of huge interest. Some of the biggest traits that we study even now with different methods. But I just want to give you an example of how these quantitative traits can be used for linkage studies. So here on slide 71 is just a list of possible quantitative traits that you might be interested in. We could come up with many others. And if you go to slide 72, what the whole idea behind quantitative trait analysis using linkage methods is that if there is familial resemblance in the levels of a quantitative trait, with the assumption that there is a quantitative trait locus gene underlying that trait, then we would expect that the IBD sharing at nearby markers would be strongly correlated with the sharing in SIBs of a particular quantitative trait. What that means is that two siblings might have very similar measurements for a particular quantitative trait and be more likely then to have sharing of a particular gene if it was actually linked to that trait. So the best way to show this is to show you an example of what's in slide 73, which is the Haseman-Elston method. This was just one approach. It's still kind of the most standard approach because it's very straightforward in how it works. But the idea here was that siblings with a high IBD sharing at a marker near a putative quantitative trait locus would be phenotypically more similar than siblings who do not share um, share who are not IBD, and they would be phenotypically less similar. So we can use for a quantitative trait, you can use a squared SIB pair difference as a measure of phenotypic similarity. And I'll show you in the next slide what that means. And then if you happen to look at a genetic marker that is very close to a gene that is really controlling a quantitative trait, then you will see a specific pattern, and that would be evidence for linkage. So let's go to slide 74 because this is the best way to illustrate it. Here we have an example that uses the Haseman-Elston algorithm. And let's just say that this is blood pressure. Let's call this blood pressure. And on the x-axis is the squared sibling difference for the two blood pressure measurements. So if you take a look on the left and we're looking at a genetic marker where the siblings are sharing two alleles, so that is like full on sharing. They're sharing two, and we see on this graph that they're sharing two when their difference between their blood pressures is basically zero, which means their measures of blood pressure are identical. So what you would see is very, very similar phenotypic measures. Here it's blood pressure in the situation where they are sharing the most genetic information. And then you can see where the biggest squared difference is, where we see no sharing. So if you wanted to use the Haseman-Elston algorithm for a whole bunch of genetic markers, and here we're just looking at one genetic marker, and you wanted to look across a lot of siblings and take the squared SIB difference for every SIB pair for that quantitative trait and then plot it with the IBD sharing, this is the kind of thing you would see if the pink boxes, that line, if there was evidence for linkage. Basically, they're sharing a lot when the difference between each SIB for that quantitative trait is basically negligible. They're very similar, and they're not sharing at all when the quantitative trait that you're interested in is very different. So this is how linkage analysis can be used for a quantitative trait. Okay, so I will talk more about genetic linkage analysis in the age of whole genome sequencing, which is a paper that I'm highlighting here on 75. I'll do that with you in person because I couldn't give this lecture in person. And I'll just end with slide 76 in saying that the things you really want to think about from this lecture, the basic concepts that I want you to understand, are things as listed here, which include what actually is linkage analysis, what is a recombination fraction, how is it defined, how is it measured, and what are the differences between parametric and non-parametric approaches to linkage analysis, and you should be able to do calculations for each. And what is a genome screen for linkage? What are considerations for interpretation? And is there more to learn from linkage studies? This is something we'll discuss in person. The answer is yes, but there are some caveats. So understanding the calculations that I described is really important. And affected SIB pair analysis, what's IBD, why is it different from IBS, how do you calculate a LOD score, phased versus unphased, what does that mean when you go to calculate a LOD score, what do you have to do, you have to account for that uncertainty. You'll be getting a homework assignment that will require you to make those calculations and get some practice, and you will see how doing these calculations by hand really helps you to understand the basic principles underlying linkage analysis, which I have to argue is just such an important topic for you to understand. We've barely scratched the surface in this one lecture, but you will have some foundational knowledge that will be important and will carry you through for the rest of the semester. Okay, and slide 77 is a PED file structure, and we're going to do that in class. Okay, thanks.